Alright, today we're going to talk a little bit more about prokaryotic cell structure and function. In class last week we talked about the cell wall and the glycocalyx, and now we're going to talk more about other aspects of the prokaryotic cell, specifically the membrane and some motility aspects. Alright, so start with the cytoplasmic membrane. So the cytoplasmic membrane in a prokaryote is actually very similar to the cytoplasmic membrane that is in our cells, our eukaryotic cells. So we still have a phospholipid bilayer. So you've seen this before. The fluid mosaic model that you've seen for eukaryotic cells still holds for prokaryotic cells. So we're going to have our phospholipids that are going to come together to form this bilayer, where we have the hydrophobic region in here with the fatty acids, and then you have the phosphate, the polar charged hydrophilic heads of the phospholipids that are on the outside, either outside of the cell or also on the inside portion of the cell. But in between that, we have our fatty acid bilayer, our fatty acid portion in the middle. And so if you look at the cytoplasmic membrane, we don't just have lipids, we also have proteins. So we have integral proteins, these are the ones here that extend all the way through the membrane. And then we have peripheral proteins that are located on, kind of attached to the membrane, but they're not actually extending through the membrane. And so the purpose of the cytoplasmic membrane is for selective permeability. So as you can see, we have this nice barrier that's only going to allow in the kind of compounds that we want inside the cell, and it's not going to allow in the ones we don't. There are some compounds that can diffuse or cross the membrane going through the lipid bilayer. Not a ton, but there are some. Other compounds that we need inside the cell that we can't get inside the cell are going to come in through the integral proteins that we have here. So the integral proteins are used to transport molecules inside the cell. Another aspect of the membrane is to set up a concentration gradient and an electrical gradient. So some of the things that can come into the cell, it can come down, we use a concentration gradient. So if we have a higher concentration of a particular compound outside the cell compared with inside the cell, we can actually allow that compound to flow down its concentration gradient inside the cell. And that is actually going to release energy that can be used by the cell. So we have a concentration gradient and an electrical gradient. I'll talk about the electrical gradient in a second. And the last purpose of the cytoplasmic membrane is for energy storage or energy production. So the energy producing compounds, and we'll talk about those when we talk about metabolism, are found in the membrane. Alright, so let's look at the electrical gradient here a little bit. So here we have our cytoplasmic membrane, and if you look at this, you can see that outside the cell, in the cell exterior, we have a higher concentration of positive charged compounds, we call protons out here, and then inside we have more negative charges. And so this electrical gradient, where we have a difference in charge, positive outside the cell, negative inside the cell, is going to be really important when it comes to producing energy. And so you can see that this gradient is set up, and we'll talk more about why that's important when we talk about Chapter 5 and energy production. All right, so flagella. Flagella, we talked about those on the outside of the cell. Flagella is used in motility. That's the major role of flagella. Not all bacteria have flagella, but a lot of them do. A lot of the ones that we work with in lab will have flagella. And so basically the flagella is just a really long structure that comes from the surface of the cell and can be used as a cell for motility. So it can use this flagella in order to move around. Flagella has three parts to it that you should know. We have the filament, which is the hollow tube made of flagellin protein. This is the part that you actually see extending from outside the cell, the filament. And so it doesn't look hollow from the outside, but inside it's really a hollow tube. So it's just a tube that has flagellin protein all the way extending around the tube, but inside it's hollow. We have a hook that's going to connect to the filament, and the hook is going to connect the filament to the third part of the flagella, which is the basal body. And so the basal body is going to anchor the hook and the filament into the cell. Okay, and so here's an example showing the three structures here. So here we have our filament. This is showing in a gram-positive cell. Here we have the filament that extends outside from the flagella. We have the hook that connects to the filament. And then we have the basal body, that's the portion seen here inside the actual cell wall. Same thing for our gram-negative cells. So we have a filament, our hook, and a basal body. The only difference between the two is that the basal body in gram-negative cells, because we have the extra outer membrane in gram-negative cells that we don't have in gram-positive cells, the basal body is a little more complex, as you can see. Alright, so there's three main types of flagella arrangement that cells can have. There's actually more than three, but there's three main ones that you can categorize flagella arrangements into, the three most common ones. So some cells, like this one seen here, have what's known as peritrochous flagella. So peritrochous flagella is when you have flagella all around the cell. So you can see we don't just have flagella in one spot or a couple spots. All around the cell we have flagella. So cells can have more than one flagella, and a lot of the really motor organisms do. And so we have a lot of flagella extending around outside the cell. You can see all the flagella coming up from the cell wall. We also have single polar flagella. So we have some cells that only have one flagella that's only located at one, we call the poles. So flagella are usually seen in rod-shaped organisms, and so we have the poles here. This is considered the pole of a rod-shaped organism. Here's one pole, and here's the other pole. And so we have the single flagella that just extends from this one pole. Or we can have tufts of polar flagella, where you can have organisms that have flagella only at one part of the cell, so only at one pole will they have flagella, but they'll have more than one type of flagella, more than one flagella there. So this organism, as you can see, has about 3 different tufts of flagella, all extending from one pole on the cell. So one of the kind of oddball bacterial shapes that we talked about last week were the spirochetes. Remember, we have the rods and the cocci, which a lot of things can fall into one of those 2 categories, but then we have this kind of odd spirochete-shaped organisms that kind of make up this odd classification. So we have these spirochetes, basically have this kind of spiral shape we see here. And so organisms that have this, the organism that causes syphilis and the organism that causes Lyme disease, have this kind of spiral shape to them. So these organisms, they don't have flagella that extend outside the cell, and so they have what's known as an endoflagella. And so what this endoflagella is basically something that's known as an axial filament. So all throughout the spiral-shaped cell, they're going to have this axial filament wrapped around the entire cell. And what this axial filament does is it actually allows them to be motile. So spirochetes are still motile, this is not in the way that we see with other organisms. And so this endoflagella that's inside the spirochetes, it's going to rotate in order to cause for motility to occur. And when it rotates, that's actually going to cause, as you can see here, we have an axial filament. When it starts to rotate, that's going to cause the spirochete to rotate as well, and it's going to move forward in kind of like a corkscrew does, like a corkscrew going into a wine bottle. You're going to have the spirochete kind of rotating and moving forward. And so it does move in very much a similar way as other cells do, except its flagella isn't outside the cell. So it kind of moves in more of a corkscrew fashion, which is a little different, but still allows for the motility that other cells can have. Right, so let's talk about the motility that cells with a normal flagella that extends outside the cell can have. And so for the motility used by organisms with a flagella outside the cell, they're basically going to have rotation of the flagella. So here's our flagella that's in the cell wall, and when this flagella rotates, that's going to basically propel the bacteria through the environment. So you can see from the arrow here, we're going to get rotation of the flagella. So rotation of the flagella, that's going to cause the bacteria to propel through the environment. And this is a fast propelling. So this is very, very fast, the rotation and the propelling of bacteria through the environment. It's very quick. And so the process of this is going to be powered either by protons or also by sodium-powered pumps. So basically what I mean is, remember we had that electrical gradient that I mentioned with the cell, where we had a higher concentration of positive charge compounds or protons outside the cell compared to inside the cell. We can use that electrical gradient, so those positive charges that are in higher concentration outside the cell, those can actually be used, to flow back into the cell down their concentration gradient. And when this happens, that's going to release energy. And the energy that's released from this is going to power our flagella, so it's going to start to rotate. So when protons flow back into the cell, that releases energy that rotates this flagella filament, which allows the cell to be able to move. And so this rotation is reversible. So you see here we have this arrows kind of going in the counterclockwise direction. That's one possible rotation, and that's the rotation that's actually going to allow the cell to move forward. But this is a reversible rotation. So this counterclockwise direction we see here, that's what's known as a run. That's going to be the forward where bacteria is actually moving towards something. But rotation in the opposite direction, so if this rotation were to turn and go in this direction, the clockwise direction instead, that's what's known as a tumble. A tumble is a much more random movement, not a movement forward, but a much more random movement where the bacteria is not really headed towards anything in particular, it's just kind of tumbling around. So these combination of runs and tumbles are both going to be important for bacterial motility. So one thing to keep in mind with this is that an organism can only run for so long before it has to tumble. So we can run, which is a forward, considered more useful from a movement, moving actually towards something for a while, and then it has to tumble. So there has to be this combination of runs and tumbles. So let's take a look at an example. So here we have our organism with its peritorchis flagella coming from the cell. And so first thing, we'll start out with the tumble. So peritorchis flagella all over the cell. These flagella are going to start rotating in the clockwise direction. So they're going to move it like a clock does towards the right, so the clockwise direction. Each flagella is going to individually rotate in the clockwise direction, and that's going to be a tumble. So here we have a tumble, our organism was here, and when it tumbles, now it's over here. Really hasn't gone in any one direction. It's just kind of randomly moving about, not really headed anywhere. After a tumble, now we can actually have a run. Now, when an organism with paratrochus flagella runs, what's going to happen is all the flagella are going to be clustered together on one side of the cell, and they're all going to rotate together in the counterclockwise direction. So all our flagella have come together and are rotating in the counterclockwise direction, which is going to allow this organism to now run. And so this run is going to propel the organism forward. So now we're actually moving somewhere. We went from here, now we're actually moving in a forward direction over here. Again, that can only go for so long before now we have to tumble again. So now we get kind of random tumbling movements. So each flagella individually rotates. Now we're randomly tumbling. We went from here to here, didn't really head any one area. And then we can run again. And again, we run, we move in one forward. And so this whole concept of running and tumbling is important for the idea of what's known as taxis. So taxis, not taxis, but taxis, is a movement in response to a particular stimulus. And so chemotaxis is when organisms are going to move in response to a particular chemical stimuli. You're going to move towards a chemical stimuli if it's an attractant, or they're going to move away from a chemical stimuli if it's a repellent. And we're going to talk about that in class more, how that process actually works. And then phototaxis is kind of like chemotaxis, except instead of moving towards a chemical stimuli, it's moving towards light stimuli. So some organisms that are photosynthetic will actually move towards light stimuli. You also have seen phototaxis. If you've ever seen plants, you have plants when they're growing by a window, they always want to grow toward the window more. They grow towards where the sunlight is. So you've actually seen phototaxis, even if you don't realize it. And so in class, we'll actually do an activity looking at how motility functions with chemotaxis and how organisms are actually able to use chemotaxis to get towards molecules that they want to be towards, how they actually can use chemotaxis to get towards things like nutrients that they actually want to use. We'll also talk about two other components of the cell, the fimbriae and the pili, which are going to be important for motility as well as for forming attachment to surfaces. So talk more about the attachment to surfaces. Let's start talking about biofilms. So biofilms are basically this organized layered system of bacteria all attached to a particular surface. So it's basically a massive growth of all of bacteria, sometimes the same type of bacteria, sometimes different types of bacteria, all attached to a surface. And so how they're attaching to the surface is through what's known as fimbriae, which I'll talk about more in class, and also the glycocalyx. Remember, the glycocalyx was that sugary, sticky substance attached to the surface around the outside of some cells. They can use this glycocalyx to attach to surfaces in order to form these biofilms. And so this sounds disgusting. The biofilms are basically a community of bacteria kind of encased in their own slime. And so here's an example of a biofilm here. So this is showing kind of scanning electron micrograph image, that's what the SEM stands for down here, of bacteria growing on a piece of medical tubing, which is kind of disgusting, yes. But you can see how we have all these different cells, so all these cells, these yellow and blue here, those are all different types of bacteria growing on this piece of medical tubing. So we have a lot of cells here that are kind of encased themselves in this community on this piece of tubing. Here's another more grim example here. This is actually showing tissue after a hip replacement that's been infected with cells growing in a biofilm. So here in the middle here, this is a mix of microbes and also pus. Pus is basically your body responding to the microbes and trying to eliminate it. So we get this kind of encased community where they grow in a biofilm on this infected tissue after the hip replacement, which is unfortunately something that happens very commonly. You can get infections when you have medical procedures done where it gets infected because it was not particularly clean or 100% clean when it went in. This is becoming a much bigger problem in today's day. So let's look at the formation of biofilms. I don't need you to know every single step in the process shown in this image here. This is actually an image from a different book, but I kind of like the way it shows the growth of the biofilm. So the first step in the biofilm is that we're going to have cells that are going to have to be attached to this some sort of a solid surface, sometimes known as a substratum. And so the substratum is going to have been preconditioned with molecules that are going to allow the cells to attach. And now here come our cells that are going to have some way of attaching to the surface. And the cells get deposited on, they're going to absorb to the surface. Some cells are going to stay there, others might desorb. So biofilm is it's a constantly changing process. You'll have cells that will attach and leave. So what's going to happen is these cells that absorb and then stick around, they're going to produce what's known as an EPS, or extracellular polymeric substances, which basically is going to be, this is what creates kind of the slime encasing. They're going to secrete a lot of sticky substances that are going to allow other cells to attach to these surfaces. And you'll see that once these cells are attached, then they're going to start secreting things. They're going to start signaling to each other, which is going to help secrete more of this EPS. So they're going to secrete, or say, hey, we're all here, let's get this party started, secrete these substances, which is going to allow the other cells to start attaching. So we're going to have transport other cells. You can see we started with a couple here, and now we're growing more and more. And so you can see this kind of cloudiness here. This is the EPS showing the slime encasing. And we have all sorts of different cells present inside this secretion. Nutrients are going to be transferred into the biofilm from outside. We're going to have kind of diffusion of oxygen and nutrients in. Each cell is going to replicate and grow, which is going to add to the size of the biofilm and more secretion of the EPS until it gets quite large. Eventually, we're going to have detachment and erosion somewhat, where pieces will detach off. There'll be cells in here that eventually will start to die and detach, but overall, once you form a biofilm, it's a very strong substance and a substance that's very difficult for us to actually eliminate. And one thing to keep in mind is that this process can be very rapid. So going from just a couple cells attached to a surface, all of a sudden a full biofilm formation can be an extremely rapid process. And when it comes to the medical community, that's actually a very big problem. So let's talk about the danger of biofilms. So this is kind of a striking number, but 99% of bacteria that are in nature are existing as a biofilm, which basically means that a lot of what we do in lab isn't always an indicator of how they're actually growing in the real world. We don't use a lot of biofilms in lab to study bacteria, but in nature they are existing as a biofilm. They're existing not only as themselves, but also existing interacting with other organisms in a biofilm. This is a huge problem for several reasons. The most obvious that we hear about most frequently would be medical problems. So 2/3 of all the infections that a human has caused by bacteria are caused by organisms growing in a biofilm. And so some of the obvious ones, such as if you have an infected tissue after a hip replacement, and also less obvious ones, such as gum disease or even just a cavity, that's caused by a biofilm growing on your teeth. And so you probably felt biofilms before. If you ever forget to brush your teeth in the morning, you get that fuzzy feeling. You know what that kind of feels like. And also certain organisms, so if you have an infection in the lungs, particularly cystic fibrosis patients that have lung infections, that's also an example of a biofilm. Industrial problems is also a huge issue. We don't think about that as much because it doesn't affect us as directly, at least not that we realize, but there are a lot of biofilms that form in industry that can be a problem and cause a lot of economic loss. So a lot of corroded pipes can occur because the biofilms are formed. If you have water filters that get clogged, those are all due to biofilms forming and basically breaking down components that shouldn't break down yet. So it causes a lot of economic loss, having to pay for things that we shouldn't have to pay for yet. Antibiotic resistance is a huge issue when it comes to biofilms. So organisms that grow in a biofilm tend to be more resistant to antibiotics than organisms that do not. And there's really two reasons for this. One is if you think about organisms in a biofilm, particularly the ones that are kind of deep in the middle of the biofilm, they're protected from the outside. Things that try to attack the biofilm from the outside aren't going to be able to get all the way down into the middle as easily. So it's kind of this physical barrier between them ever even being exposed to the antibiotic when they're growing in a biofilm. Second reason why organisms in a biofilm are more resistant to antibiotics is that they actually express different genes in the biofilm. So certain organisms in a biofilm are going to express genes that make them more resistant to antibiotics than other organisms. So we get kind of this difference in gene expression and also this physical barrier. So anything that can grow in a biofilm is more capable of being resistant to antibiotics than anything that is not capable of growing in a biofilm. Now, it's not all doom and gloom, of course. We know with microbiology there's always a good side, so there are some beneficial uses to biofilms. For example, wastewater and sewage treatment. So if you go to wastewater treatment plants, they actually use a biofilm of organisms. The organisms that grow in a biofilm are actually able to break down the components and clean the wastewater and sewage, so we can use it to our benefit. And also bioremediation, which I talked about last week with the current event. When we talk about bioremediation, that's basically an example of a biofilm. So when you want to clean up a chemical spill of some sort, you want to have an organism that can actually break down that chemical, you have to grow them as a biofilm on top of that chemical in order to break it down. And so it's important that we study biofilms because the more we understand about how biofilms form and how they function, we can actually use them to our advantage to prevent other biofilms from causing a problem or to fix the problem that we ourselves cause with industry.